want to just review for the audience the data for bispecific antibodies, or BiTEs. Slightly different technologies, but we'll lump them together for today. Peter, you've had some experience with some of these. Do you want to talk us through that?
Sure. So at LCI, we've been involved in the phase 1 Janssen study of teclistamab. This is a BCMA-targeted bispecific antibody. So basically what this is, it's a kind of two-pronged antibody as I describe it. So one prong of the antibody binds to BCMA on the surface of plasma cells, and then the other prong of the antibody binds CD3 on T cells and activates T cells, basically creates an immune synapse between the T cell and the myeloma cell, and engages that T cell, to destroy the myeloma cell. So basically the blinatumomab, if you will, of multiple myeloma. And there's several different products that are out there. There's AMG 420, which was kind of the first proof of concept, of BiTEs in this specific space. The problem with the AMG 420 was that it had to be administered as a 4-week continuous infusion, much like blinatumomab has to be administered. These next generation BCMA bispecifics have a longer half-life and can be dosed less frequently. So in the teclistamab phase 1, the bispecific antibody was administered with step-up dosing during the first week and then weekly therapeutic dosing thereafter. And my colleague Saad Usmani presented this data at ASCO, and at the higher dose level that they looked at with the IV formulation, they're seeing response rates at this point of 67%. Granted, a very small number of patients, 12, but the patients that I've treated on these things have very refractory disease, extramedullary involvement, and I'm seeing really quite impressive responses and durable responses with this agent.
And that seems to be a common theme to all of the BiTEs. We have many of them in clinical trials now, at least 6. So not to pick on any specific one, but I do know that Tom at ASH, I think you presented on a different one from a different company, but tell us a little bit about the results of that. And maybe since we haven't covered it yet, the toxicity.
Yeah, so we have been part of the Celgene trial for their bispecific T-cell engager, and it was presented by Dr. Costas at ASH. They really also did a first-in-human dose escalation trial, but when they treated 9 patients at what they believe is going to be the dose they're going to take to an expansion and then to a phase 2, which is a step-up dose of 6 to 10, and then some patients at 10. So 9 patients treated at that higher dose. They had 8 out of 9 responders, and 5 out of those people were MRD negative. So pretty impressive, actually. So response rate almost 90% for these nine patients. I have a few patients on it, and they, when patients get on it, it appears durable. The big question in the waterfall plot that was shown at ASCO for the teclistamab, there were patients out 19 months, and most of the patients that are out 12 months are still continuing in response. So the question is, will it be as good as the CARs or not? And I think we're all really looking forward to seeing those data. This might be easier. The problem is it does require hospital stay. The toxicity is the cytokine release syndrome, the CRS. It requires a hospital stay for the first dose and every step-up dose because you might activate more and more T cells each time. But after you're done with the step-up dosing in the first real dose, then the T cells, then there is essentially no CRS. And so patients may need to be hospitalized once, twice, or 3 times before they can continue, but then can continue in the outpatient clinic thereafter. The neurotoxicity is also similar, but much less. Less than 10% neurotoxicity. You still do see some cytopenias. About a third of the patients do get cytopenias. But like you said earlier, this is a really heavily pretreated population. Maybe it'll be less if we do it in less heavily pretreated patients. Of all the agents, these are my favorite.
Well, let's come to the advantages and disadvantages in a second to close out, but I just inject my own experience. We've used a couple of these agents targeting different companies, different agents. My impression is they're a lot less toxic than CAR T cells, and I suspect could probably be delivered safely as an outpatient from the majority of patients, particularly if you had some home monitoring system that you could use. I think also, to go back to CAR T, I suspect for some of those products, that's on the horizon as well, that they may not require hospitalization for everybody, at least initially. The only thing I've been a bit concerned about is, unlike CAR T, at least in my experience, the remission duration has not been as long, and that the people do progress more quickly, at least in my short experience. So I think that's something you have to pay attention to. But off the shelf, don't have to wait, good high response rates, and I think obviously going to be a very important addition to our therapy.